So the last talk was a really good talk. See what I can do here. This is my group, and a lot of this work has been done, some by John Quay, he's now at Stanford as a postdoc. Keopan, he's now at MIT as a postdoc. Enrique over here, he's a professor in Chile, so a bunch of different people contributed. I'd like to start out just mentioning the grand challenges in global health, and I'm sure you've all seen that, but it's really good for a general audience. I'll talk a little bit about P epitope, how this is a predictor of vaccine effectiveness. And then I'll talk a little bit about virus evolution. I'll put that in the context of an epidemiology model. And then I'll move to detecting new flu strains. And then if we have time, I'll talk a little bit about dengue. So usually when I talk about vaccines, I like to show this, that 6 of the 14 grand challenges in global health are related to vaccines. So vaccines are important. And then for Andrew here, influenza and pneumonia, just to put people sort of on the same page, yeah, it is a pretty significant cause of death. This is just a personal story. I was traveling from Paris to London through the super tunnel, the channel, whatever it's called. And when I got to London, there was this mask wafting along on the ground. I'd never seen that before in a Western country. And this was in, let's see, this must have been in Paris. This was during the swine flu, right? And somewhere on here it says avoid contact with people in public places. And this is in the train stations. Doing my best. This came out the day before I had a talk at BARDA, which is the part of the U.S. sort of sits like in between the CDC and FDA, I guess. They write the contracts to the vaccine manufacturers. So there was this nice thing about the swine flu. Maybe the mortality a little bit higher this year, and then later down in the article it said, it's possible that the attack rate will be very high this year. Maybe it'll be 40%, maybe 50%. So keep that number in mind. I'll come back to that later. So again, this is just a history of the strains that we've been infected with over time, starting, at least we know, 1918 with the H1N1, and we heard about the swine flu in 1968. What the immune system tends to recognize are 2 proteins on the surface, right? Hemagglutinin and neuraminidase. And I'll talk a lot today about immune recognition of hemagglutinin. And there are 5 regions that people have sort of agreed are recognized by the immune system. There's some discussion what that actually means. And I'll focus on these 5 regions, these epitope regions of HA. So this is sort of an overall view of the talk, that influenza is recognized by the immune system, primarily by antibodies binding the hemagglutinin. And this then puts a selective pressure on the hemagglutinin to evolve. And this looks a lot like the figure you've seen before. I'll tell you exactly what this is. This is sequence data, not ferret data. And there's this evolution along this curve in this 2-dimensional space as a function of time. This is the evolution of the flu over the last 40 years. So saying it again, there's this pressure on the flu virus to evolve. The virus falls away from this pressure, and really we'd like to understand at various levels of detail, either from sequence data or maybe all the way down to atomic detail, can we calculate what that pressure on the virus is? And then these are figures I think that have been shown already. Basically, we used to die from a lot of stuff in the past, but now we're much cleaner. But there is this seasonal pattern, and some of that is definitely influenza. And I never noticed it before, but this is from your 122 cities. So again, I'll focus on the hemagglutinin, right? And so I'm mostly focused on the H, and I'm really focused on these 5 epitope regions of the H. And I'll mention a few times during the talk this order parameter. So I figure out which of the epitopes is dominant. So the concept is which of the ones of these 5 epitopes is most dominantly recognized by the immune system. And then I see how different that dominant epitope is between 2 strains. And that difference I call P epitope. It's the number of amino acids that differ divided by the size of the epitope. That will turn out to be a pretty good predictor of cross-reactivity. It's actually better than the ferrets. Okay. And this is a tough challenge, right? Because you have to pick in February, March, what we're going to get in October. And the process for making this is slow, and it's not just the making of the vaccine. So BARDA was telling me during that swine flu thing, they had a lot of vaccine in July. It just wasn't filled. It wasn't into the single use or the multiple use vials. So the filling is a limitation. There are only a few fill lines, and that's actually maybe more serious limitation than actually making it. So this will be maybe a little bit controversial, and I don't always necessarily say this anymore, but honestly, this is how I got interested in the flu. I got a flu shot at Costco near Los Angeles a long time ago, and the nurse told me if I got a flu shot that year and not the next year, I might be more likely to get the flu the next year than if I hadn't gotten the flu shot that year. Well, I don't know about that one. That seems kind of weird. So how can the shot make you more susceptible? You know, normally vaccination lasts a long time, and it's surprising that vaccination against the flu could make you more susceptible to the flu. So we thought about that for about 5 years and published our first paper 5 years later. I'm a theorist, so what I wanted to do was come up with a theoretical model of the immune recognition of the vaccine and then the subsequent response to the virus. And I'll show you, we'll get to data, don't worry about that, data's coming. So from a theoretical point of view, this is sort of how a condensed matter physicist, how a theoretical physicist would think about this. We'd like to make a spin glass model because we think immune evolution is relatively difficult. The idea is that your antibodies fold, they need to fold, but they also need to function, and the evolution of those antibodies, really the B cells that make the antibodies, that occurs in this complicated but correlated landscape. So I won't say too much about this, won't worry about that too much, but that's the model that I'll be using to generate theoretical curves, and then I'll start to compare those to some experimental data. So we need to know more about the immune response. We know that the immune system makes these antibodies. The antibodies are made by VDJ recombination, right? So you have several different V regions, several different D regions, several different J regions in your genome. Those are chosen with some probability, combined together to make your initial set of diversity, your naive set of antibodies. And for any given antigen that you are attacked with, you about 1 in 10 to the 5 of your antibodies will tend to recognize that. You have about 10 to the 8 different antibodies, so you'll tend to respond with about 1,000 different sequences to any particular antigen. And those then will undergo rounds of mutation and selection, right? We know roughly how quickly B cells divide. We know roughly what the mutation rate is per division. Rough response period, you know, maybe 7 to 10 days. And we know what the binding constants are. So the best of your naive antibodies has a binding constant around 10 to the 4. After the primary response, it's about 10 to the 6. After a secondary response, it's about 10 to the 7. So we can use 10 to the 4 and 10 to the 7 to start to fit some of our parameters, and then we starting to have some agreement with data here with this 10 to the 6. So in the beginning, I was very focused on this, what you'll learn I call original antigenic sin. Now I tend to say really this is just a model that in general will tell us the effectiveness of the vaccine. But let me just say a little bit about the original antigenic sin. There are 2 ways you could get something like this original antigenic sin. So one possibility is what I call localization. So here is the ability, it's the free energy, so it's kind of the inverse of the fitness. It's the ability of the antibodies to recognize the antigen in sequence space. So sequence space is highly dimensional. This is just an artist's one-dimensional slice through it. Basically saying, okay, there's some good antibodies here that have a low free energy of binding. Next year, the flu will be a little different because it's mutated. So the region of good antibodies will be somewhere else, but there might still be a peak. There might still be this good region from last year, and there may be a barrier in between. If that's the case, then you'll have these memory antibodies next year. These would have been the best, but you'll have these memory antibodies, and they'll a little bit interfere with what your naive antibodies would have evolved towards. So this competition from the memory antibodies can be one reason to have suboptimal immune response. Another possibility is, I said you start with 1,000 sequences, but just to make a picture, let's start with 10. Those don't actually find the best antibody. They find very quickly sort of a local optimum nearby in sequence space. And if you start with 10, you're going to find a better local optimum than if you start with 5. And this happens in the immune system. There's something like a conservation of mass in your immune system, and if you use more memory sequences, you're going to use fewer naive sequences. They will be expanded. So this would be like half of your response was memory, and then the other half was naive. That means you have less diversity you're using of your naive repertoire. So I'll show you that both of these things happen. There are a few more numbers we need to know. I said you have about 10 to the 8 different antibodies, but you have the ability to make about 10 to the 11 different heavy chain variable regions. And then this is something, of course, that we have to build into the modeling, right? We want the model to have that same set of genetic diversity. Okay. And then another point is this binding constant. So the binding constant is telling you how much antigen is bound to the antibody. And the amount of antibody binding is proportional to the antibody concentration. It's also proportional to this binding constant. And that's important because the binding constant evolves over time, right? The somatic hypermutation improves this binding constant. Also, the concentration of antibodies increases over time, right? So the concentration goes from naive to the effectors. That's about a factor of 1,000. And then it comes back down. So maybe it's only 100 times higher, and then that slowly decays over months to years. Okay. So here are some dynamics that may make what I've been saying a little bit more clear. We have a person. This person is vaccinated against a given strain. And actually this is averaged over many people. That's why it's smooth. Then let's say the person sees the same thing. So the person's vaccinated. The person's immune system improves, does the somatic hypermutation, and then we wait a while. Maybe wait till next year. Or we wait until later in the season. And then the person sees the same thing. If the person sees the same thing, the immune system will actually just continue on improving those antibodies a little bit better. If the person sees a somewhat different thing, the person will use some memory sequences and some of those naive sequences from the random pool and evolve from there. So what we want to do is compare the response in the presence of the vaccine, which are these numbers, to the response in the absence of the vaccine. And the response in the presence of the vaccine depends on how different the vaccine is from the virus. So that's what I'm showing here. I'm taking these curves and I just shift them to the left, and then I plot one of these that I shifted to the left with the original curve. So here's the primary response, and then here's a secondary response for one particular value of the distance between vaccine and virus. And initially, the memory sequence looks a lot better than the primary response, but it evolves a little bit more slowly. It's a little bit stuck. It's a little bit more clear down here. Initially, the memory sequence looks better than the naive sequence, but after 10 days, it's actually a little bit worse. So that means there's a little bit of a barrier around those memory sequences. They have a little bit harder time evolving out of their little area than the naive sequences do. So this will make it a little bit more quantitative. I want to compare these secondary responses to the primary response as a function of how different the vaccine and the virus are. So that's what I'm doing here. 0 means the virus is the same as the vaccine. So the response here is better than the response there. 1 means the virus is quite completely different from the vaccine. It's like you're vaccinated with tetanus and then you see the flu. There should be not too much cross-reactivity. So, okay, there's no difference. It's 0. In between, the vaccine is less and less effective. And there's this region here where, because you got the vaccine, you're making a little bit worse response than you would have if you had not had the vaccine. Here are the same data plotted in terms of binding constant. So this is a typical primary response, 10 to the 6. If the vaccine is the same as the virus, that's a good vaccine. You make a better response because you had the vaccine. So this is where most childhood vaccines are, right? Measles, mumps, rubella, they don't evolve much. Those are good vaccines. Out here, there's no correlation between the vaccine and the virus. Here, the virus is mutating. There's a distance between the virus and the vaccine is increasing. And of course, then the vaccine's getting less and less useful. And we'll look at the data later, but at least here predicted is this region where you're more susceptible to the virus because you got the vaccine. So we could either focus a lot on that and get worked up about that, or more generally we could say, well, we have sort of a theory of effectiveness of the vaccine. And there's something interesting there, but in general, we have a theory of the effectiveness of the vaccine. Let me say a little bit more about the immune response before I show you some data. There are some generic, interesting properties. So we've heard a lot about cross-reactivity. Cross-reactivity is sort of a biochemistry experiment. You take the antibodies that were produced in response to a given antigen. You see how well they recognize some other antigen. So that's not an immune response, right? You're not allowing the B cells to evolve anymore. You're just seeing how well do those antibody molecules recognize something different. And sort of generically, those primary responses, they're going to have a 10 to the 6 binding constant. And if you change the antigen, usually, because that antibody was optimized somewhat for that particular antigen, usually they'll be worse for any other antigen on average. And usually that decay is exponential. And experimentally, cross-reactivity ceases in the range 0.3 to 0.4. That was measured in terms of fraction of amino acids being changed. And that's usually defined as the binding constant going below 10 to the 2 or so. So in the theory, that occurs at about 0.36. So that's pretty good. 0.36 is in the range where we expect to see this cross-reactivity to cease. So not only do we get the exponential decay, but also do we see the cross-reactivity ceasing at roughly quantitatively the right number. We can explain that exponential decrease in infinity. Basically, when the flu, we quantify the distance between the virus and the vaccine in terms of how different the model is. So essentially we have these terms in the model, and when they change, we end up with only a fraction of the energy being common. And that's what, when you exponentiate it, that's what gives you the exponential decay. Mutations are a key aspect of the immune response. And this is the number of mutations in the primary response, about 9.3. There were 30 rounds, and the mutation rate was a half. So if there were no selection, the mutations you would observe would be 15. But of course, there's selection, right? The antibodies are being selected because for their ability to recognize the flu. So there's an observed substitution rate, I guess I should probably really call it. Now, during the secondary response, if the virus is the same as the vaccine, the immune system won't make another 9.3 mutations because these were already pretty good. It'll make fewer. It's starting to get stuck. So it turns out it makes around 7. The number of mutations rises. Now, if the virus is completely different from the vaccine, then that's just another primary response, 9.3. In between, the curve rises. It rises above the primary response substitution rate. And this is roughly the region of that original antigenic sin. So that peak I was talking about between the two valleys, that peak is sort of entropic in nature. The immune system's having to work harder to get from where the memory sequences are to where the naive sequences would go. Okay. And then the last sort of general point is diversity is another key issue. So here's the diversity of the naive repertoire that does recognize the antigen. So these are the 1,000 sequences. As the somatic hypermutation process proceeds, those improve. This is in some energy scale. Those sequences improve, and the best of them come out to here. Now, you don't actually make the best antibody, right? You make a distribution of antibodies, and you make maybe 3 to 5 at reasonably high concentration, and they'll have energies out here. Now, we can look at cross-reactivity. So let's say we change the antigen by, let's say, a half. Then what your immune system will tend to do is, in the secondary response, those memory sequences have energies that are around here. So our immune system will probably use all of these naive sequences. It'll probably use those memory sequences, and then it'll try to improve that set. So you notice what happened is the memory sequences basically cut off the tail of the naive distribution. That's what I was saying before. If you use memory, you're therefore going to throw out some of your naive because it looks less promising than your memory. And the immune system will only spend a certain amount of, there's only so much mass that the immune system is willing to spend on this process. So we can kind of quantify that. What happens if you throw out the tail of the distribution? So if you don't throw out any of it in the primary response in these energy units, this is what I've been showing you all along. If you throw out everything except best one, that doesn't work very well. So if you do this very strong goal seeking, you get stuck sort of pretty quickly, and you don't make as good a response as if you keep more diversity. If you throw out half, in this energy scale, it's worse by about 0.25, and that was roughly the height of that original antigenic peak in energy units that I showed you. So we're seeing reduction of diversity, and we're also seeing localization in the immune response. So now I want to start to make connection with data. And to summarize what I've said so far, this theory that I briefly mentioned, it's a form of a random energy model or spin glass model. These are widely used in physics to describe disordered systems. Really, it describes a rugged fitness landscape. And then, of course, there are correlations, though, because we know that antibodies are made by this VDJ recombination process. So it's not completely uncorrelated. There are correlations there. Mutation of the virus corresponds to parameters in the model changing. And in the immune system, well, I mentioned people believe that there are these 5 epitopes, right? And I postulate what the immune system sees as change is the fraction of amino acids that change in the dominant epitope. So that's a postulate right now on this slide, and we can see how well that postulate does. Here, this really should be vaccine effectiveness, not efficacy, but this was in 2006. We didn't completely know. So this is vaccine effectiveness as a function of that order parameter, P epitope. And there's no reason, okay, sorry, so the symbols, those are data that a really good undergraduate went through the literature to find. There's no reason they should fall on a line, but this is just, the dash is just a linear least squares fit to the data. And then the solid, that is the theory that I described for you. There's one free parameter in the theory, which is the point here. And I set that to be the average of the efficacy when the effectiveness when the virus is the same as the vaccine. So for example, this is not a free parameter. That's a property of the theory. So this first point is the theory is reasonably good agreement with the data. From now on, let's forget about the theory. So then the second point is this order parameter, P epitope, this postulated thing, it actually correlates the data pretty well. So how do we quantify that? R squared is about 0.81. Here is vaccine effectiveness in humans as a function of one or another antigenic distance defined from ferrets. Correlation is not too good. So ferrets can give you for a short distance or a big distance, give you what in this case was not a very effective vaccine in humans. So we know animal models are not perfect. That's not surprising. It's a little surprising the theory does reasonably well, actually even does a little bit better than the animal models. So that's kind of cool. Yeah.
Brief question. You put together a lot of data from different sources.
Yeah.
So
let me show you the error bars.
I'll just amplify a little bit, maybe. What you call ferret data is actually antisera made to a particular virus in a naive ferret model. So that is how well...
I think it's ferret HI data.
That's right. That's how well an antibody raised to a particular virus reacts to a virus that would come from a human, for example.
Yeah.
That's different than the human response.
Definitely is.
And that's not what
and no one... It is what I showed.
Is that what you showed in the...
The y-axis are human responses.
Okay, so that...
No one reports human HI data. Japanese used to, but they don't anymore.
Right. Well...
So I'd love to use human... I'm not saying this is best one could do, but this is what's usually available.
I haven't read the paper, but I think we should talk because there are groups, including FDA and Canada, that do have some human data.
You used to report...
Fairly
so the FDI...
limited amount of data, but that is collected over time.
Yeah, so
it's part of the strain selection procedure now, as you know, for flu vaccine selection criteria.
You don't really use HI data from humans for that.
Yes, we do.
They're not even... So we asked Nancy Cox about this repeatedly.
My fault.
She knows me. Human HI data were occasionally reported in the FDA strain sequence reports, but not all the time and not recently. So recently, for whatever reason, there seem to be fewer human HI data available to the public than in the past. Japanese used to report more, but they don't seem to do it anymore. So we asked Nancy, why is that? My recollection is that she said, well, that didn't seem to help all that much, so we didn't use it. So we could either, I may be misrecalling what she said, but it's true that there are fewer human HI data now than before. And you can find many recent FDA reports from the strain selection that have no human HI data in them.
The last 2 years will be
Good.
But anyway, that's a side conversation.
So these are ferret data.
So on the right is ferret data,
The
right?
x-axis, can't see because it's cut off, but there are two ways typically antigenic distances are defined from ferrets. Either you take a log or you take a square root. Those are the two x-axes, and the y-axis is effectiveness in humans.
As age 5.
U minus V over U. So effectiveness is something epidemiologists define.
This is not what an epidemiologist would call, that's my other main point. This is not what an epidemiologist would call efficacy or effectiveness.
I think this is what an epidemiologist would call effectiveness.
No, it's not.
So let me say what U is. How can you say it's not? What's U?
Well, go ahead, tell me what U is.
So U is the rate at which a person who is not vaccinated gets infected. V is the rate at which a person who is vaccinated gets infected. And U is the rate at which people who are not vaccinated get infected. So if vaccinated people never got infected, efficacy would be 1. If the vaccine had no impact, efficacy would be 0. You don't think that's effectiveness?
No, I do. I think that.
So,
Well,
there you go.
There are very limited opportunities where you would have that data combined with virus.
Well, we worked pretty
From the
hard.
People who were studied. I mean, we've actually been trying to get that data for the past
This
5
is-
or 7 years or so.
You're going to hate my next statement,
Okay.
and it's related to what you just said. This is ILI.
Say it again.
It's ILI.
It's ILI. Because
Then there's the problem.
Because-
Okay, thank you. Now I understand.
that's why these are lower than you would have expected. Because, as you say, there's so little lab confirmed, and we'd love to have that, right? So give it to us.
No, doesn't quite work that way.
There you go. All right. So, coming back to evolution, there's pressure on the virus to evolve, right? The virus will evolve away from this pressure. The pressure is from either vaccination or from prior exposure. So what can we say theoretically about that? If we recall what I just showed, let's not worry about this negative stuff. Let's say that the effectiveness of the vaccine, it goes to roughly 0 at a value of about 0.19. So that means the virus will be feeling pressure from either the vaccine or prior exposure when P epitope is less than 0.19. So there's pressure on the virus to evolve a distance of 0.19 or greater. And if you look at, when we look at the distance between the vaccine and the virus in that year or in the next year, let's do the next year, on average, so this is averaged then over all sequences in GenBank or GIS-AID, it's about 0.16. So the virus is evolving on average about that far. So almost as far as we would predict. So that's interesting. I'm going to go towards an atomistic calculation because there's one case where we can do that. Before we did the atomistic calculation, we had this sort of hand-waving argument. And the concept is, okay, let's imagine we have a crystal structure of an antibody binding to, say, the Hong Kong strain, because that actually does exist. Let's pretend that antibody is representative of the antibodies in the population, and that may or may not be true, but let's just pretend that. And what is the virus going to want to do? The virus's fitness will be increased when it makes mutations that decrease the binding with the antibody. So those will be selected for. Mutations that increase the binding with the antibody will be selected against, because those viruses will be killed off. And we're going to calculate that change of free energy in a second. But here on this slide, we're going to make a hand-waving argument, which is one of the ways to decrease this affinity of binding is you just put more charge on the surface of the virus. If you put more charge on the surface of the virus where the antibody is binding, the virus will become more hydrophilic. It will go away from the antibody and towards the water. And the referees didn't believe that, so this actually came out second. I'll show you quantitatively that that's true. Here, I'll show what you can explain if you believe that argument. What I'm plotting is the number of charged amino acids in the epitope region of the virus. This is for the Hong Kong strain. And so it came out in 1968. Epitope B was dominant. Here are the number of charged amino acids in epitope B. Then epitope A became dominant, and the number of charges in B go down, right? Because now there's a little bit of drift. But the number of charges in A go up. A was dominant. So that's kind of interesting. We can model that. We can try to fit a model for a stochastic transition matrix, basically, to those data. And that stochastic transition matrix is quite different than the one that you would use for typical protein evolution. So it looks like there's selective pressure on the virus. Not surprising, right? Looks like there's pressure on the virus to run away from the immune system. You can explain it specifically in terms of the composition of the epitope region. Here are some guinea pig experiments done by my collaborators, so not by me. And again, basically, these are basically just showing you can see a little bit of evolution in a guinea pig experiment. You can see 2 to 3 to 4 substitutions. And the substitutions are ones that tend to lead to an increase in charge.
This slide has 2 graphs.
This slide has 2 graphs. Yeah, so I think one was when we, so my collaborators got the vaccine strain from CDC. It's actually a mixture. It's got more than one thing in it. And I think one of these graphs is from as delivered from the CDC. And one was where my collaborators purified it to just one strain.
Every shipment of influenza virus contains what could be called a selection of quasi-strains, because just the point you mentioned, the polymerase of this particular negative strand RNA virus is very messy.
Plus growing it in the eggs introduces some stuff.
So every time you get, that's why when CDC, for instance, describes the virus that goes into this vaccine strain selection procedure, it's not A Wyoming 2003. They will say A Wyoming 2003 -like.
So I may say something about this likeness
because there's
too.
always a quasi-species, and it actually, if you look at it, it actually makes a very nice normal curve of the virus that you will get out of any particular person, or out of any particular cell culture, or out of any particular egg isolate.
Yeah.
Can I just ask just a quick?
Yep.
Sorry, going back one.
Yep.
Is there a cost to having more charge? Because you said
There is.
it comes
Yeah.
down from drift.
Yeah. So this
But
must.
in drift, you'd expect it as much to go up as.
This is
down.
because of the cost.
Yeah, yeah. So what's the cost of the?
So let me show you some numbers.
Okay.
I'll just wave my hands even more. So.
Can I ask a very,
Yep.
very practical question?
Yeah.
What's the citation on the bottom of those 2 slides that we can't read?
Sorry. JME 2011, volume 72, page 90.
And we're going to try to fix that projector. Well, he's going to try it right now. Whoops, don't fall.
Intervention's coming now.
Oh, no. It's
No.
not.
It's in a connection.
Empiricism.
Not ours. It's deep.
So for this one particular case where there is a crystal structure of the antibody bound to Hong Kong, then we can actually do real calculations. So my background is statistical physics, so this we shouldn't have had to do this. And basically what we're trying to do is see how the binding constant changes as we change the amino acids on the surface in this epitope region of the virus. And there's some technical tricks. There's this Hess cycle that thermodynamics people love. There's actually something that we noticed the way so physical chemists should have been able to do this for 30 years, these transmutation type of calculations. There's actually a small mistake that everybody's been making related to the endpoints that we fixed with some Einstein crystals, which represents about 40% of the free energy. So there's a small detail there that's actually important, so we were happy to figure that out. In the end, okay, you can get numbers for, I'll show you what these mean in a second. So right now this is just a picture. You can get numbers for how does the free energy change when we mutate to different when we substitute to different amino acids on the virus. So here, for example, is showing that one of the four most disruptive things that can be done, these are the four highest substitution free energies. And these are free energies, right? Not energies, they're free energies. So this is what reality is. This has entropy in it. The four most disruptive things that can be done are to put on a charge. So this hand waving we had, it's actually true. So we were happy with that. So this actually came out first, and then we confronted the referees of JME with this. They had to relent. So now let's look at some human data, because we have sequences, right, of the Hong Kong, and we can see what were the substitutions and which ones actually fixed. And now a couple points here. The substitutions tend to be towards amino acids that are disruptive. So the virus is trying to run away, and when I say that, what it means is that there was selection for the virus to be running away from this antibody. It doesn't make the mutation to the most disruptive amino acid because there's entropy here, right? It doesn't have to make, it doesn't benefit tremendously by making the most disruptive substitution. And that's very rare to find the most disruptive substitution. It just finds a somewhat disruptive substitution. So there's an entropy or a finite size effect here that one should keep in mind. But we do see that these substitutions that are occurring in humans, they seem to be the ones that are a little bit more disruptive. If we really take that calculation and push it, let's assume that the fitness of the virus is proportional to the change in free energy. That's a pretty strong assumption, but let's make that assumption. And then here are predictions of how long it takes for a substitution to fix. And here's what happened from the sequence data. So, the time scale, it's off by 25% or so, but not too bad. So that was a long, complicated, a long, detailed calculation, and it does maybe provide something useful.
Just to ask a quick naive question about
Yeah.
this in some area, but so some of these, you're assuming a particular antibody, right? That's sort of fixed.
We're not assuming a particular antibody. There are very few structures of antibodies bound to viruses. There's like one almost
Okay, so there really
bound. There wasn't much of a choice.
Okay.
We're assuming that that's representative of the population, and it's kind of hard to believe that's a very good assumption, but that's the best one can. So I'm going to go on to something else. But if we want to do this level of detail, you have to have a structure.
Right, because the question I had is this, that you're just saying, okay, I can put charge on this and this will cause it to run away from the antibody. The question is, are some of these mutations more, can the immune system more easily respond to some of these mutations than others? Because you might imagine I have something where you're using strategy A, and I can counter with strategy B, but you have a very easily evolvable counter to that strategy. So in that context, if you're looking at the population level, even though it is an immediate response, this best response would seem very good, you never see it happen, because as soon as I try it, you whack me down. And so what will actually evolve may be things that look suboptimal as the best response in the short run, but are better evolutionarily because of the response on the other side. But I don't know what the human
So
side
that's
is.
actually a pretty deep question. Let me answer it in a trivial way first.
That's fine.
One of the things you said was, well, some substitutions might be more useful for you than others. So this P epitope, it's defined just as the fraction of amino acids that change. And that sounds like there's like no chemistry in there, there's no biology in there, there's almost nothing. So you should be able to do a more careful definition of distance. We tried 4 or 5 different things. None of them really seem to be any better. So actually, this P epitope is saying, well, just running away to anything is it's already a very large jump, and it doesn't really matter what you mutate towards. However, you are asking a pretty deep question. So one of the things I would say the virus does is put these epitopes out on its surface, and there's some cost to that, but it allows the immune system to recognize it, and there's kind of a co-evolution here. And then once one epitope is too well recognized, there it does the epitope switching, and some other epitope becomes dominant. So there's clearly a cost to the virus to do that, but there's a long-term benefit. So that's a little bit more deeper answer to your
Right,
question.
and just to add, what can happen is constrained, because what does HA do? Why don't you tell the audience what HA actually does?
Yeah, so one of the things HA does is it has the silac acid binding site at the top of it. So there should be some constraints around that binding site. And if we looked
That's
very
how
hard,
the virus gets into our particular respiratory epithelial cells, whether we're people or chickens or whatever, has to bind to the sialic acid.
yeah. So if we look,
That's the constraint, if you will, on the way that the virus can evolve.
yeah, it's only on one of the epitopes, but if we looked very hard, we should be able to see that. You should be able to maybe make an even better P epitope incorporating that constraint, for example. Okay, now I'm not an epidemiologist either, but let me just briefly talk a little bit about that. So we saw pictures like this, right? So there's a distribution of humans, 7 billion, we heard, right? And at long scales, they are moved around by air, right, on planes. But then within a city, there's person-to-person contact. So there are at least 2 scales of contact patterns, and this is how the virus moves around, right? So let's make a stochastic model here where we have groups, and then we distribute these groups around the world in cities according to a certain city size distribution, which looks like this. And then we also have the transportation between the cities. And within the US, we can get better data than we can for the world. So we end up using, again, one of these distributions to tell us what the flights are between the cities. And then here, I think this is the exact same plot that was shown in the last talk, although I have 2005, you had 2004, so I don't know what the deal is with that. But these look, I looked at yours, it looks exactly the same. It looked different up here, but it looks exactly the same down here. So this is distribution of within city networks. So we put that into the model as well. So we'd like to use something that's measured, but for the time being, well, we'll just use this distribution. And here are WHO data on isolates. We shift those and average over years. That gives us the crosses. And then this line, of course, there are a bunch of parameters here, but this line, this curve, then is what the model I just described very briefly tells us should happen. So basically, you see the outbreak occurring in November, December, and sort of coming down in January, February. This theory does more than that, though, right? So it doesn't use an input R0, it predicts an R0, right? Because it's based upon these more fundamental parameters. So I didn't mention this, but in this model, you have these individuals. Individuals have an immune history. And that, so it's not just SIR. They have a history of, say, up to 100 previous exposures. And the immune recognition is based upon that P epitope theory that I showed you. So starting from these maybe more basic immunological things that we feel we've shown, then we can predict things like R0. We can predict viral diversity compared to data. I think that was from Ferguson also. And then we can predict things like, I don't know if this is really efficacy or effectiveness because it's a prediction, but maybe it's efficacy. We can predict how efficacious is a vaccine if you vaccinate 10% of the population, 40% of the population, 70% of the population, right? Of course, 70% is better, but how much better? And then if the vaccine and the virus differ by a certain amount, how does that reduce the efficacy? If you have a single component vaccine versus a multi-component vaccine, what happens? So if you have a single component vaccine, as the virus runs away from it, the vaccine will become less effective. But if you have a multi-component vaccine that covers the distribution of viral, the quasi-species, then the vaccine is efficacious against the virus. Here's showing sort of the inverse, it's just the attack rate. So we can put numbers on this. And then my student defined population at risk. So this is, of course, well known in risk analysis and finance. Here, we're defining population at risk as within some error bars, what is the largest fraction of the population that will be infected. And the kind of cool thing, remember what fraction of the population it was said might be infected by the swine flu? It's like 45, 50%. Well, that depends on your, can't see it here with this projector, but that depends on your error bar, right? So here's 94% confidence interval with a single component vaccine. It's like 30%. If we go to 98% confidence interval, the single component vaccine, it's like 50%. So sure, yeah, maybe 50% of the population will be infected if you want a 98% confidence interval. So we can be precise about what we mean there. So the last thing I want to talk about was clustering to detect strains. So we've always been asked for at least 5 years, can you predict what the new strain will be? That's really hard because the space of possibilities is really big. And really, the virus just has to run away from current recognition. It can run away in any direction. So I think that's really hard to predict, but we can detect it. So what do I mean by that? So this looks like antigenic cartography. It's using the same standard dimensional scaling as antigenic cartography, but it's not using ferret data. And I already kind of showed some graphs where maybe ferrets are not that predictive. Or you could say, well, there are more sequence data available. So that's what this is using, sequence data. And the idea is, can you describe the strain evolution in sequence space? You project it to the two most informative dimensions. That's just standard dimensional scaling. And then you see what you get. So here's the same figure. We did this first for the H1N1. There was a Texas strain and a New York strain. Those of you who work in this will know about. They're actually very similar, though. So although we can see two clusters, I haven't told you what the scale means yet. These are really small distances. So to our immune system, those are the same.
This is all hemagglutinin?
This is all hemagglutinin, yeah. So we originally worked a little bit on neuraminidase 2. It wasn't really all that predictive. So we focus quite a bit now on hemagglutinin. So again, we take the sequence data. We do this multidimensional scaling to find the two most informative dimensions, a standard statistical technique. And that'll, if we see a new peak growing, so if we see a new peak, that's interesting. We want to see that new peak growing. So it has to be spreading out. If it's not spreading out, there's no chance it'll be the peak next year. And then third, we want to see that that peak is sufficiently far from the current peak that the immune system won't recognize it. So for the pandemic H1N1, this is actually a very short distance. So as of when I made this, that was not a new strain, really. So let me show you something more interesting. That same year, when everyone was worried about H1N1, there was a strain of H3N2 that emerged. It was first sequenced on the 15th of March. It was not considered a new strain until somewhat later, of course, right? And we have some documentation on this if we want to get into details. But of course it takes some time to detect it, right? So what can this clustering thing do? This clustering, it's really impossible to see here, but good, this one. This is as of the end of March. So 15 days after it was first sequenced, we see this new cluster growing. And we can detect new strains with about 10 to 12 we can detect new clusters with about 10 to 12 new strains there. So we see this thing growing, and this is what it looks, of course, a few months afterwards. And this didn't infect many people in the northern hemisphere, but in some southern hemisphere countries, it did infect 20 to 30% of the people. And then the next year, this became the dominant H3N2 strain. So we can see clusters fairly quickly after they first emerge. Here's another example. This was from the Fujian. It became dominant here in 2003, 2004. Before that, the circulating strain was Panama. And we can see it emerging fairly early on. So we can see it, this is as of 9/30/2002, and this is as of 2/1/2003. And I'll have a summary of this for you in a few slides. This is another example. This is of California. So California became the dominant strain 2005, 2006. Here is as of 9/30/2004, and this is 2/1/2004. So we can start to see these strains, months to 1 to 2 years before they become dominant. So here's a systematic summary, including the thing, there's no H1N1 here, but including the two H3N2 cases, it's really everything from 1996 on. So the Fujian, we see it a little bit before it becomes dominant. The California, we see it right when it becomes dominant. So, in a couple of years we do better than the WHO prediction. We never do worse. So this looks like maybe something interesting. We cannot predict what the new strain will be if there are no data, but from the data that are currently available, so GenBank or GIS-AID, we can detect the new strains. So that's kind of interesting. And then let me briefly just mention dengue fever. I'm not a virologist, but I got interested in dengue fever because I read if you're infected with one of the subtypes, there are 4 subtypes, you are more susceptible to the more serious form of dengue, used to be called dengue hemorrhagic fever, from the other 3 subtypes. And presumably the same thing is true for vaccination. So you want the vaccine to contain all 4 subtypes. Now, just some background, dengue, it's been called the most important mosquito-borne virus. About 2.5 billion people live in the 100 or so countries in which it is endemic. 50 to 100 million people get infected with it each year. It's a pretty unpleasant disease. It's like super serious high altitude sickness, basically. And there's this secondary form, used to be called DHF, that is even more unpleasant. I mentioned there are these 4 serotypes. Here are data from a vaccine trial in Thailand in humans, and what this in red is showing is those data averaged. Basically, it's showing the immune response to a 4-component vaccine. These serotypes are recognized pretty well. These, not so much. And I couldn't tell you looking at that this vaccine isn't good enough, but yes, this vaccine is not good enough. So what that means is this recognition of those strains needs to be made higher. So the first thing we did, so this is called immunodominance. Some of the serotypes are more dominantly recognized than others. So the first thing we did was try to explain that. Immunodominance is pretty mysterious in the literature. And our explanation, so one possible mechanism, is this is competition for access to antigen. And your T cells will recognize some antigens more than others. Your T cells are selected for, right? They don't evolve, but they're selected for, so there's obviously competition. And, well, some antigens are going to be more recognizable than others. So just naturally, you're going to recognize some antigens, you're going to make more T cells against them than others, if there's competition for access to that antigen. With a fitness landscape described by the spin glass model I showed you, that's the open bars. So it's pretty good agreement. There's sort of one adjustable parameter here. There's pretty good agreement between clinical trials and theory, much better than we should really expect. But the nice thing is that theory gave us an idea. So I said it's competition for access to antigen. That competition occurs in the lymph nodes. Humans have hundreds of lymph nodes. So what if you vaccinate with serotype 1 here, 2 here, 3 here, 4 there? You might make good T cells against serotype 1 here in the draining lymph nodes, 2 here, 3 here, 4 there. So theoretically, that's a really good idea. You can't see it at all, but the very non-uniform response, here is almost equalized. So it ends up looking sort of like that. Experimentally, do I not have that slide? Experimentally, there are, this is a little old, there are 3 proof of principle experiments, 2 in mice on HIV. So if you vaccinate with 2 epitopes in 2 different places, you get a broader response than the 2 in the same place. If you vaccinate with 3 clades in different places, you get a broader response than 3 clades in one place. There was actually a monkey experiment with dengue, which wasn't a great experiment, but it did show you get a broader response with the multi-site vaccination. And there's a Sanofi patent, which cites this idea in their dengue vaccine. So that's kind of interesting. Yeah, I didn't talk about the first one, but the second conclusion is this p epitope. Well, so we have this whole nice statistical physics theory behind it, but just forgetting all that, p epitopes correlates pretty well with vaccine effectiveness in humans. Second, these novel dominant strains, we can detect them months to years before they become dominant, so that would maybe be helpful to authorities. And then third, this multi-site vaccination, it appears to alleviate immunodominance, certainly in the theory, but then even in a few proof of principle experiments. So with that, thank you for your attention. I'd be happy to answer any more questions.
Michael, when you found the Fujian strain sort of incipiently,
Yep.
Do you know from GenBank if you found it in Japan? Or
Uh,
I guess it's China, pardon me.
I don't know standing here, but yes, that if I went back to the data, we would see that because it usually has the location of the sequence.
So that would be interesting to
Yeah, so to really put this in geography would be interesting. Yeah.
When did they stop calling it dengue hemorrhagic fever? Why do you
Like a year or two ago. I forget what it's called now. Dengue shock syndrome, yeah.
But that's just that's more severe. That's just a more severe outcome from dengue hemorrhagic fever. That's not.
Well, but even,
It's immune response. Severity of illness to dengue is moderated to a great degree by our immune response to the virus.
Right. I'm just.
so when we wrote our paper in 2006, we could still call it dengue hemorrhagic fever, but in the last year or two, that seems to be discouraged.
Huh, that's interesting. I mean, all the surveillance systems I work with still call it dengue hemorrhagic fever.
I don't know. These dengue guys, what can I say? They know more than I do.
Your third point about the prediction of new strains.
Yes.
You mentioned that it's much better than what the WHO does. What
I was careful not to quite be that strong.
okay.
It offers additional information.
What they do currently.
Sorry, say that again?
Yeah, what approach WHO takes to make their
So it's a combination of, so I think the first pass is just sequence, right? Make phylogenetic trees. And then the second more expensive pass is to go through the ferret HI studies. And in some past years there was HI data used from humans, but I don't think that that's always the case.
And that's incorrect, actually.
Which one?
So there's actually 4 sort of groups of studies that go into the WHO vaccine strain selection procedure. The first is correct, it's just sequence data. And until we know, we don't have a map between sequence and antigenic characteristics that are meaningful for changing vaccine. Until we have that Rosetta
Well,
Stone,
that's what I showed you.
sequence-only data will never be the gold standard.
Well.
So that's not there yet. But so the first is correct, is sequence. The second step is HI data, and there's a ton of HI data because Mike's correct, it's fairly inexpensive to get. And the way that data typically is presented now, he mentioned this, is the work from Derek Smith and Colin Russell called antigenic cartography. So there's a way.
Yeah, but that's not used really
That's
extensively.
Been to the meetings, you haven't. It's
I talked to those
absolutely
guys.
used. It's absolutely used.
Derek wants it used more.
He wants it used more.
That means it's not completely used.
Absolutely used. Strain selection.
It is partially used, yes.
Used. The third is, as you said, there's limited data, but there is some. So human serology data. So a particular person is vaccinated to a particular strain, that antisera is used to react to the viruses that are being isolated across the world, and we see how well it makes sense. And the fourth bit of data, which is the most sort of intensive to get, I don't think you mentioned, is virus neutralization data. So HI, hemagglutination inhibition, is mixing red blood cells that have sialic acid receptors on them with virus, putting in an antibody that will prevent the viruses from agglutinating if it reacts to the particular flu virus. Flu viruses have the ability to agglutinate red blood cells. There's all sorts of technical details in terms of types of blood cells in some years and some strains. Guinea pig red blood cells work better, for example, than blood cells from turkeys.
Yeah.
But there's a lot of HI data because it's relatively inexpensive to get. But the final, the most meaningful data in terms of virus data is actually how well antibodies can neutralize a particular virus, and that means neutralize their ability to cause infection in a susceptible cell. So there's 4 sort of broad groups of data that go to the vaccine strain selection.
So let me push back a little bit. You said the sequence data is just not there.
No, I didn't say it's not there.
You did
No,
say
I
it's
didn't.
not there.
No, I said
It's pretty good.
There's no gold standard to understand how well.
It's pretty good. People want to maybe not acknowledge it, but it's pretty good. It's not bad. So there's this, that's capturing 81%. And then there was the clustering. Actually works pretty well. Never worse. It's better in 2 cases. It's actually pretty good.
Yeah, but this is. We can talk more. I mean, this is.
You disparage it, but it's actually pretty good.
I'm not disparaging it. I'm saying that this is. This is looking backward, right?
Right, so we want to do this prospectively now, a couple
Exactly.
years.
Exactly.
Yeah.
So no one, I don't think anybody would disagree that the current capability to get a lot more sequence data is very meaningful for the vaccine strain selection procedure. But it doesn't,
People used to say with the current data,
in and of itself, it's not not everything. Sequence is not everything. 'Cause
Also-
we have to understand, as you would agree, I think, we have to understand what a meaningful change is in sequence.
P epitope.
So there's more than one epitope.
Yeah, there are 5. Yeah, so P epitope is the change in the dominant epitope. People used to say, you know, that old sequence data, there just wasn't enough. You couldn't see new strains. So a lot of people didn't think we could do this. Actually, even with that old data where there wasn't so much, even then, it still works reasonably well. So it certainly works better now with the more data, but it's not bad.
Can you explain, like, for those of us who don't work with these data, like, so do you like get an email from GenBank, you know, by some automated Deem?
No, we're not that intense. Basically, it's when when we decide, you know, really to get down to it and do our final figure, we download everything as of that date, and you'll probably find a footnote somewhere in the paper, you know, GenBank accessed on whenever, GIS AID accessed on whenever. So we ourselves will not update our database internally. We'll access the master database as of whenever we do this analysis.
But I mean, every few weeks during GenBank gets an update or whatever, and you can
Daily. I mean, you know, all the time they're getting updates.
and then you could recalculate.
So yes, one could, for example, I think that would be maybe most interesting in the context of these figures. So these, I picked the most recent one, like this one, you know, you could do this in real time every week. You could redo this and you could see, well, is this thing growing? Is it not growing? Yeah, that'd be kind of cool to do. That'd be very interesting to do. It's a very,
You do like a cron job, right? I mean,
Yeah.
in theory.
it's a very sensible suggestion. You could make movies. Any other questions?
Back to the discussion about predicting, Nancy
Yes.
Cox and Blank had a paper that made quite a splash, what, 5, 6 years ago? Maybe it was about predicting, looking at the trees. Is that the bell, or am I not remembering?
I'm not remembering what that paper was in terms of prediction.
We'll talk about
I
it.
think you're probably talking about one of the science papers.
Yes.
That used antigen cartography.
Yeah.
Well, yeah, Robin Bush.
Right.
Yeah.
Robin is here, right? Yeah.
Yeah, she's here, but she's, wasn't able to come to our place.
Right, and Rob-
But she's here, yeah.
Yeah, so,
So how does that paper fit in?
right, so there's some nice data. I'm not sure if I've got it hidden. There's some nice, I didn't talk about H1N1 at all here, but we can do all this for H1N1. Let me see if I can even find that citation. So we looked at evolution rates of the virus. And that paper by Robin had evolution rates in it. Yeah. Let me see where that would be. I'm going the wrong way. So there are rates of evolution of H3N2 and of H1N1. And in the old days, everybody said H1N1 evolves less quickly than H3N2. If you normalize it by how frequently H1N1 or H3N2 are dominant, then you get a little bit different conclusion. So for something like this, so we're looking at evolution rates of H1N1 or of H3N2 normalized per dominant season, and then H1N1 actually evolves a little bit faster than H3N2 does if you're counting evolution only during the seasons in which it's dominant, in which it has pressure on it, really. And I guess I'm not citing that particular paper here, but in our paper we do cite that Bush one because they had some of these numbers in there. So it turns out that the H1N1 vaccine, it has more cross activity than the H3N2 vaccine does, if you just look at the epidemiological data. So I mean, we were all worked up about that swine flu, but not only were there fewer deaths than average, actually the H1N1 vaccine is better than the H3N2 vaccine. It's more cross-reactive. So I didn't know that in the beginning, but that's why we did this study. And that then led us to, if you really seriously believe that, that means there's more pressure on H1N1 than H3N2. So H1N1 should be evolving more, which is not what people thought. But well, most of the time we're not infected. It's not the dominant species, not the dominant strain, so there's no pressure on it. So if you normalize by number of dominant seasons, actually, yes, H1N1 does evolve a little bit more than H3N2.
Speaking of evolving, we're evolving close to the time when
Yep.
the shuttle is going to be picking you up.
Good.
So I think we're going to have to thank our speaker.